Hello, my name is Kathy Shair. I'm an assistant professor in the Department of Microbiology and Molecular Genetics. I'm also affiliated with PMI, or the Program in Microbiology and Immunology. Today, I would like to share with you a short presentation with some slides showing some of the active projects going on in the lab. My lab is located at the Hillman Cancer Center in Shadyside, only a short distance from the main campus. We belong to a program known as the Cancer Virology Program, where commonly multiple investigators are interested in understanding the underlying molecular mechanism of how viruses can cause human cancers. The virus that my lab studies is called Epstein-Barr virus, where it is associated with both B-cell lymphomas and epithelial cell malignancies. EBV is actually quite a common pathogen, and you're likely to already be infected with it. More than 90 to 95% of adults worldwide have already been infected with EBV, carrying the virus for the host's lifetime and continually shedding and transmitting it to others via your saliva. So what we're interested in is why some individuals might be prone to EBV-associated cancer. Now, I mentioned that it is associated with various different types of B-cell lymphomas and epithelial cell cancers. But the main focus of our lab currently is in a head and neck cancer known as nasopharyngeal carcinoma, where there's approximately 80,000 new cases of NPC, or nasopharyngeal carcinoma, arising each year. And almost all cases from endemic areas are associated with clonal and latent infection of Epstein-Barr virus. The lab is broadly divided into three major projects that are grouped into cancer mechanisms, EBV molecular virology, and in NPC biomarker discovery. In terms of cancer mechanisms, we study how two principal EBV oncogenic proteins known as the latent membrane protein 1 and latent membrane protein 2A sometimes interfere with DNA damage repair mechanisms and DNA damage response signaling. And so there we're studying with very unique nasopharyngeal carcinoma cell lines and other non-tumorous nasopharyngeal derived cell lines, how the expression of these two proteins or the absence of them in the presence of EBV latent infection changes the outcome of a cell exposed to a genetic insult. One of the most exciting aspects ongoing in the lab right now is we've recently developed very new 3D cell culture models in order to study the very many diversified cell types of the nasopharyngeal epithelium, which is composed of a stratified epithelium as well as a pseudostratified airway epithelium. These are extremely new models that are unique to our lab that we've been able to develop uniquely at Pitt because of our collaboration with the Cystic Fibrosis Research Corps and the Department of Otolaryngology. The third research project is focused on using liquid biopsy as a form of informing whether we can predict nasopharyngeal carcinoma risk. And this is important because nasopharyngeal carcinoma often arises with peak incidence in males in their 50s. And so this is critically important for obviously identifying individuals at risk and hoping to treat them early, as well as for testing new candidate EBV vaccines. And so here I'm just displaying an example of how complex these signaling proteins are that emanate from these transmembrane oncogenic signaling proteins. And so we're largely focused on deciphering how these protein-protein interactions with host signaling protein pathways end up interfering with the host cell signaling mechanism, as well as how this ultimately benefits the fitness of EBV. I just want to spend 2 slides showcasing these brand new 3D cell culture models. Here is an example of the techniques we use that are very specialized in terms of looking at the molecular virology as well as by imaging to section the nasopharyngeal pseudostratified as well as the stratified epithelium. In this slide, we're just looking at the pseudostratified 3D cell culture models generated from nasopharyngeal cytobrush scrapings of someone's nose. In this case, that's COVID-19 negative. And so here we can see examples of the expression of the main EBV epithelial cell receptor known as ephrin receptor A2, and different cellular markers that mark the different types of cells, ciliated and mucosal secretory cells, showing successful differentiation of the pseudostratified epithelium. We go on to infect these with EBV and profile how different donors may respond differently or have different susceptibilities. The second aspect is we've learned that EBV replicates to extremely high levels in the stratified epithelium, which is most similar to the types of cells you know on your skin. Here is an example where we stain for a lytic protein known as zebra, showing abundant expression of this lytic protein. And we use specialized imaging software to score for these staining foci. Furthermore, we can do specialized EBV molecular virology assays, in this case, staining for EBV DNA in a technique known as fluorescence in situ hybridization. Really what I wanted to show you here is these are extremely new and exciting times for us using these 3D cell culture models, because historically, almost all of those studies have been done in 2D cell culture, and they physiologically do not mimic, authentically mimic a large aspect of the nasopharyngeal epithelium. What we've also begun to do is to look at the transcriptional profile, which cannot be tackled by conventional bulk RNA-seq methods. And so we have embarked on a journey to use a new technology known as single-cell RNA-seq, because the many cell types represented in such 3D cell cultures can only be discerned or properly discriminated by single-cell sequencing. And so, to my knowledge, we are one of the first labs to be able to analyze EBV infection in the nasopharyngeal cell cultures as well as in vivo. And here we use different methods in which to transcriptionally profile the different cell types using cellular marker genes, as well as looking into each cluster of cells for evidence of individual EBV transcripts. And you can see an example of some of our work in a preprint article listed here in bioRxiv. So I think these are generally quite interesting times for us with the 3 different projects that are quite special, require some quite specialized techniques involving imaging and molecular virology. And we couldn't do any of this without our key collaborators, both inside Pitt as well as some people outside of Pitt. And here, this was just the last image I have of everybody sitting together without a mask from December 2019. And these are the current lab members, and I've also listed some of the past lab members. If you have any questions, don't hesitate to email me or look for some more information from the listed websites here. Thank you.